morning, everyone. My name is Michaela. I'm a content strategist with BioXSpace, and welcome to BioXSpace. Today with us, we have Dr. Rini Chaturvedi, a researcher and a mentor. Dr. Rini is a researcher with over 4 years of experience in cancer biology, parasitology, molecular, and structural biology. She has her doctorate focused on drug discovery. She has worked at academic and industrial institutes such as the Institute of Microbial Technology, Chandigarh, Postgraduate Institute of Medical Education and Research, Chandigarh, and Daiichi Sankyo India Pharma Private Limited, Gurgaon. Her crucial work has been on screening novel biomarkers in the endometrioid type of endometrial carcinoma and therapies targeting tyrosine kinase in non-small cell lung cancer. She has academic and industrial experience working on novel antibacterial targets and discovering small molecules targeting various biological pathways in apicomplexan parasites. Today, this workshop will be hands-on on drug discovery and some applications associated with it. Over to you, Dr. Rini.
I hope my screen is visible now.
Yes, it is.
So, good morning, everyone. I am Dr. Rini Chaturvedi. I have completed my PhD from ICGEB New Delhi. And currently, I am working in ICGEB only in the structural parasitology group. And my work has been on folate biopathways enzymes found in the parasite Plasmodium. That is the organism responsible for causing malaria as such. So today I'll be talking about most of the times about protein and protein visualization tools like CHIMER, Pymol. And before that, a brief introduction about proteins and X-ray crystallography. So we know that the X-ray crystallization or X-rays were discovered by Roentgen in 1895. But for the first time, that X-ray diffraction via crystals was first discovered by Knipping and Loy in 1914. So there was a gap of around almost 80 to 90 years that people were able to understand that X-rays can also be taken into consideration for diffraction of the proteins and understanding its molecular structures. Now, XRD is X-ray diffraction is a non-destructive analytical technique for identification and quantitative determination of various crystalline forms, which is known as phases. Now, it is almost around 90% of the proteins or the structures have now been predicted by X-ray crystallography. Other techniques like NMR, that is nuclear magnetic resonance, and cryo-EM are also in use nowadays quite a lot. But most of the time, XRD is a major technique which is getting utilized. Now, it is important to understand that why we need to understand the atomic resolution or the molecular structures. So for that, it is important that accurate and precise molecular structure models at atomic resolution needs to be obtained rapidly, and they are obtained rapidly and reliably by X-ray crystallography. Identification is done when X-rays are, they are shown upon the crystals of the proteins or any kind of macromolecule, and we get a diffraction pattern out of it. Now, the theory to determine crystal structures from the diffraction pattern was developed by Bragg, and we know about Bragg's equation as well. So the figure over here, it shows that the lady of DNA, that is Rosalind Franklin, in 1950s, it was because of her work that the DNA structure was first solved by Watson and Crick in 1953. So Rosalind Franklin actually worked with Morris Wilkins, and she provided the measurements on the chemistry of DNA. And the the diffraction pattern, the most famous diffraction pattern, was given by her. Now, this work was utilized, and the structure for DNA was solved by Watson and Crick. And for that, they were awarded Nobel Prize. So first, I'll be talking about a brief introduction of how we actually utilize and bioinformatic analysis for identification of target genes, going to translation of the proteins, etc. So we need to understand what we need to actually do. So for that, we need to know what target gene we will be working on. So for that target gene, the genome sequence or the gene sequence can be extracted from many of the softwares available, which are freely available in the Google or any kind of net. So basically we can go to NCBI and select gene over here and then write the gene's name over here. We will get a page which will give all the details of the gene, and we can actually take the FASTA sequence from here, which can be either then later translated by using XPASI, or we can also check the protein sequence, the amino acid sequence from NCBI only also as well. So rather than using gene, we can actually click on the protein over here and then type in the protein's name. So it will give a amino acids for that particular organism, and there might be many organisms from which we have to select the organism which we are interested in. So if we are going for the translate tool, so we go to XPASI, we just copy and paste our DNA sequence over here. This tool, this freely available software, actually gives us the amino acid sequence in this format. Now the structure for the amino acids which we have received, the protein structure can be either done by two ways. So we can go for homology modeling if the crystal structure is not known, or we can also go for crystal structure. So for crystal structure, I'll be talking about later, but for the amino acid sequence which we have received over here, which we can actually put into various tools like iTASR, FIRE, Swiss model, Schrodinger, these are all freely available tools. For Schrodinger, the license is required only for doing a higher grade of bioinformatic analysis like docking, simulation, etc. But we do not need license for doing such simple work like, just predicting a structure based on a reference molecule. So we will talk a little bit about proteins first. So we know that there are around 22 amino acids, and they are classified into either polar, non-polar, that is hydrophilic or hydrophobic respectively, and charged and positive and negative charged. So when amino acids join together by forming peptide bonds, it forms the primary structure of the protein. When the primary structure of the protein interlinks together and formed by the use of hydrogen bonding, it either forms 2 to 3 type of primary secondary structures, that is alpha helix. When the first residue and fourth residue forms hydrogen bonding, it gives around an alpha helix, and there are 3.6 turns per basis. Whereas when the amino acids or the amino polypeptide chain is in a conformation in a parallelly or antiparallel strands, it is known as beta helix. There's another secondary structure which is known as turns. Now, when the alpha helix or beta sheets are combined together, it forms a 3D structure known as tertiary structure. We can see over here that is a beta polypeptide chains. Whereas higher structures, the tertiary structures, when they combine together to form a quaternary structure, it is shown over here. Now, when we talk about crystal structures, if we know that there's a protein for which the crystal structure has already been solved, where do we need to go and search for that particular Pdb Id? That is, Protein Data Bank is the one freely available website where all the proteins structures are annotated and deposited over here. Once any group solves a protein structure, they try to submit it to this particular website. So there's not only RCSP PDB, but there's another website known as EBI or EMBL PDB, from which we can actually isolate the structures. So what we need to do, we just need to write the enzyme's name or any structure's name over here and then search for it. So we will get that particular PDB ID. And then which we can later actually search for other things and work on other things. So this is over here. So the growth of Protein Data Bank, we understand that the protein structure is required for understanding its mechanism, its function, and for drug discovery programs also we need to understand what an enzyme or a protein looks like and how it works like. We need to understand its active site also. So there has been a tremendous growth in the position of the structures in Protein Data Bank. So since 1972, we can see over here a myoglobin structure was solved and is deposited, whereas in 2008, and I'm talking about like 10 years back, there were these many molecular structures were solved. And I'm pretty sure that there might be more than 1 lakh or 2 lakh structures which has been deposited to protein data bank on a regular basis. So if we need to know and understand how to go along with the crystallization of a protein and then solve its structure, it is important to know the basics and the steps involved in it. So we need to, as I said earlier, we need to understand the target gene required for it. We need to clone it in a cloning vector. Then we need to express it in a expression vectors. After we understand that the protein is getting expressed either in a bacterial system or a mammalian system, we need to purify it. And for crystallization studies, the protein should be homogeneous, highly homogeneous, and it should be highly concentrated as well. So the purity should be more than at least 95%. If the protein is not pure, then we cannot crystallize it, or it is a possibility even if we crystallize it, the other impurities might crystallize rather than our protein of interest. So we can see that here the protein was expressed and purified, and we get a single band that is around 99% pure and a single peak in the GPC, that is gel permeation chromatography. Now for crystallization, there is a method known as vapor diffusion method, which is the most common method for crystallization in XRD. So what happens is that the macromolecule needs to be in a super saturated form. So for that, in vapor diffusion method, the protein sample and a precipitant is sealed in a vacuum chamber box. And here we can see that this is the protein sample and this is the precipitant or the reservoir or mother liquor as we say it. So what happens is that water starts to diffuse out of it, water vapor diffuses out of it, and it tries to maintain the osmolarity of both protein sample and the precipitant together. So this leads to the dehydration of the protein drop and the precipitant as well. So this causes slowly concentration of the protein and the precipitant together, and hence crystallization occurs. Now crystallization occurs in the supersaturated form and in the nucleation phase. So we can see over here that this is the nucleation phase where crystallization occurs and it needs to be supersaturated. Moving on to data collection. Now, when we see that there's a crystal in our trays or in our vapor diffusion method chamber, we check it with a microscope and then we send it for X-ray diffraction. So when X-rays are diffracted onto the crystal, we get reflection files like this. And from this, we can actually go for model building of the refinement using various tools like Phoenix, Poot, etc. And there's a suite known as CCP4 suite, which can be used for all these things. Now, the model building or refinement for protein, basically most of the time molecular replacement method is used, because molecular replacement uses a reference molecule, which is somewhat similar to our protein of interest. So that protein of interest, the sequence for our protein of interest and the reference molecule, the identity should be at least more than 40%. So the structure is determined based on a reference molecule, which we already know, and this is the electron density of a protein. We model that particular structure based on that reference. We fit in the amino acids in the density maps, and then we get a new protein structure, the atomic model for the same. So the final structure which we get is somewhat like this, and we need to understand its all the structures, all the active site, where the ligand will bind, where the natural substrates will bind. So this is all. So now this is the SARS-CoV-2 spike protein, which was recently solved in 2020 January. And since this was solved, we were able to understand how SARS-CoV-2 is acting, and there was so much revolutionary work was done because of this only. So I have told about this. So these are the electron density map of each amino acid which has been which shows how the amino acid side chains are getting fitted into the in the electron density maps. Now, the molecular refinement and the crystal structure solving part will be later discussed in the upcoming lectures. I will be just talking about the molecular visualization tools right now. So when we check for Phoenix, this is the main Phoenix graphical user interface window, whereas this shows the Poot graphical interface window in which integration and refinement is done with the help of another software, Poot. So for molecular visualization tools, there are many tools which we use these days. We have Chimera like this. Another is Pymol. These are all freely available softwares which you can download. So when we switch on Chimera, we see this particular type of window over here, and there are so many options over here. We can actually go and open any kind of file in this. So I'll just show you how to open a file in this first. So you'll go to open. So it will give a window like this. And if you want to change the file type, right now it is PDB. It's better to check what file types can be checked in this. So yeah. So we can actually see aligned faster. We can see the CCP4 density maps, the density map which I just showed you for amino acid or any kind of protein which we can see. So density maps also come into this. And yeah. So there are many options for this. So we can go for Python as well. We can go for PDB as well. So I'll just click on PDB. And if you have a PDB ID, then you can actually click on it and save it and then go for it. To understand how to retrieve a PDB ID, we will go to Google and check for RCSB PDB. So RCSB PDB. I hope my screen is visible. Yeah, Michaela, my screen is visible, right?
Yes, Dr. Rini.
Okay, so you just click on the RCSB PDB, and then you can actually write whatever enzyme or whatever structure you want to check on and write it over here. So I will be working on DHPS enzyme, that is dihydroapterate synthase. So since my work is on Plasmodium, I will show you the Plasmodiums only. So the left panel shows the scientific name of the source organism. So there are many CPDB IDs for different organisms. So we need to understand whatever organism you want to check, you have to check it from here. So I will click on Plasmodium falciparum. You can also go for taxonomy accordingly, that only if you want to see only the bacteria's PDB IDs, then it will show over here, or eukaryotas or archaea, etc. We can also check for the experimental method. So for X-ray diffraction, there are 417 in structures which have been submitted through XRD, whereas for cryo-EM, around 126 structures have been submitted. So we can also see these refinement resolutions, whether the crystal structure which was submitted had high resolution or low resolution. So we can check it from here. The release date, enzyme classification name. There are so many options that you can check on this particular site. So I have selected Plasmodium falciparum, and you just need to click on run. So there are around 13 structures which have been submitted for Plasmodium falciparum DHPS enzyme. So this is the PDB IDs, the main IDs which we need to enter into either Chimera or Pymol. So 6JWR, 6JWS, all these are the PDB IDs of the same enzyme, but somewhat different. Like some of them are wild type, some of them are mutant, and some of them have different substrates to it. Some of them have even ligands to it. So I will be going on with the 6JWR or 6JWS. Now 6JWS is the one that is showing a mutation, that is A437G in the DHPS domain of the protein. Whereas 6JWT, there's a triple mutation over here. So I will take 6JWQ, and it is the wild type form of the DHPS enzyme. So I will go to Chimera now. You can save this particular ID here, download file, or you can also go for view file. So you need to click on this if you want, and then you can actually understand what exactly is inside the page. So 6JWQ is the PDB ID. This is the validation plot for the protein structure which was submitted to it. The paper which showed all these protein structures. Now we can see that the chains in this particular structure are two, that is A and B, and the sequence length, the amino acid sequence length is around 728 amino acids, and You can see the hydropathy plot, the disordering of the plots, etc. in this. And you can also see the ligands in here. We can see that there's ATP, there's AMP to it, there's ACT, there is Ca, that is calcium ion, there's magnesium ion, there's adenosine monophosphate. This is all the experimental data for the structure validation. Now, this is 6JWQ. If you want, then you can display file over here as well, or you can download file here, the faster sequence of PD format. So if you want to go to Chimera and open the PDB format, so you need to download it first and then open it through your system. Or alternatively, you can also do one thing. You can go to Chimera, go to file and fetch by ID. So when I say fetch by ID, you need to write over here the PDB ID, that is 6JWQ, and then fetch. So we will get this particular molecule over here in the Chimera. Now, since I just told you that there were two chains of this particular enzyme, now before working on this protein, we need to understand what this protein does. Now, this protein is is involved in formation of tetrahydrofolate that is required for vitamin D, and this is one of the proteins which is already a known target for one of the drugs, the sulfadoxine drug. Now, this drug is already in the market and it is provided for malaria patients in India and many other places as well. Now, sulfur drug also targets other microorganisms like Clostridium difficile, etc. So this is the major reason that I have selected this particular enzyme, so that we know that the crystal structure which we have is already a target of a drug which has been used for some quite time now. So now these are the two chains. Now, if you need to select one particular chain, there are many different ways by which you can do it. So if you want to select one chain or one beta helix, so you can either use a mouse as such. So you just go to select one this, you need to press Control and select. So this particular residue has been selected and this is glutamine 362. Now, if you need to select this particular alpha helix, you just select, just press upper arrow. Now the entire alpha helix has been selected. Now, if you want to select the entire chain A, then you need to press one more upward arrow. So we can see that chain A has been selected. Now, this is one way of doing it, or you can do one thing, or another thing is that you go to select, click on it, go to chain A or B. So try to go for chain A. Now, this is the chain A, which has been selected directly from these options. Now, if you want to select chain B, you will go to chain and then select B. So we can see that this chain has been selected. Now, if you want to remove the selection, you just go to select and clear selection. Or you can also do one thing. If you want to select, go to chain A. And if you're not interested in, selecting this, what you can do, you just go to select and invert all models. So when it is invert all models, what will happen is whatever the selection was, it will just go to the other selected ones and not select the ones which we have selected previously. So I will just tell you how to check one particular chain. So I will be deleting this chain. Now, if you want to delete this particular chain or any kind of residue or ligand or residue, you need to select it first. Since this is selected, I will go to actions, go to atoms or bonds, and then go to delete. So this entire chain will be deleted. So now we have a monomer of the DHPase enzyme. Now before working on this, I will also tell you that this particular DHPase enzyme is fused with another enzyme, that is HPPK enzyme of Plasmodium falciparum. Now, this particular enzyme is seen as a bifunctional enzyme and is fused only in Plasmodium falciparum and a few other organisms like Francisella, whereas in all other organisms, this enzyme exists as a separate entity. That is, HPPK will be different and DHPase will be different. So this is a complex molecule, but it's important to understand what exactly is going on. So we can see that apart from the alpha helix, beta sheets. So we can see that these are the alpha helix. So I'll just press control and select over here, and then upward arrow. So we can see over here that alpha helix has been selected. Now, if you want to see if there are any other, how alpha helix is attached to another alpha helix, we can see over here that there is a loop. So I'll just press control and then upward arrow. So now this particular alpha helix is connected to this alpha helix. So for this also, if you want to select this loop along with this alpha helix, we will press control and shift and then select it. So this will be selected and then upward arrow. So this entire loop and another small alpha helix has been selected. Similarly, if you want to check this loop, this particular long loop, what we can do, I will again press control and shift and select any of the residues. Take this, for example, and upward arrow. So this way we can select as much as we want. So there won't be an issue with that, right? So this is how you select a structure in the Chimera. Control for one single residue, control and shift for several different residues. And then upward and arrow and downward arrow, if you don't want to select it, you just press downward arrow. Yeah, so this is deselected now. So I hope I'm clear with this. Okay, so moving on to other things. Now we can see that there are many other options in the select option. So we know how to select a chain A or B. Now we can also select chemistry of the structures, that is element. We can select carbon, all the carbon atoms. Since this is a protein molecule, there will be carbon atoms everywhere. So it gets selected everywhere. Now if you want to check the H hydrogen molecule, you just go to element and select H, right? And then go for either O. We can check over here how many oxygen molecules are over there. Or we can go to the residues. Now residues are amino acid categories also. So we can check all the aliphatic amino acids wheresoever are there. Now if you need to check for the aliphatic amino acids, what you can do, it has been selected. Now if you want to change the color so that you can understand it later on, what you need to do, you need to go to actions, color, and let's color it, suppose, orange. So we will remember these are the residues which are aliphatic in nature. So we can go for functional groups like this. Then we can go for residues. If we want to understand what are the aromatic amino acids in this, we need to select those. Now this has been selected. So we can scroll on to this. So we can see at 97th position, tyrosine is present, which is an aromatic amino acid. Similarly, at 232, tyrosine is present. At 61 also tyrosine. And this is phenylalanine at 315 residue. So what we'll do, we'll go to residues. We know these are the amino acid categories, and we can actually color them accordingly, and we can actually study what ones are those.
Dr. Rini, this is all very interesting and informative, I must say.
Okay.
I think we might need to cover Pymol in another section of our workshop series, as
Yes,
this workshop times out in 50 minutes, so we have about 15 minutes remaining.
Okay, fine. So what I'll do, I'll just give you a short overview of Chimera only,
Okay.
because Pymol is very much similar to Chimera.
Thank you.
Yeah. So when we go to residues, we can see the active site. So we need to understand where ATP is binding. So you just select ATP, and we can see over here the ATP is present. Now, if you want to focus on ATP and the residues along with it, you just go to action and then press focus. So here is our ATP molecule, which is interacting with these many residues. For interaction, I don't think I have the time to show you that, but yeah, so ATP is over here. We can also go for another residue, that is, take magnesium, for example. Magnesium, this ball, small ball is over here. Since we know that ATP is required for, magnesium is required for the binding of ATP in this particular active site. We can see over here. Now I'll just clear the selection and I'll focus it. So apart from the alpha helices and all, we can see there are ligands over here, and this is the active site of the HPPK enzyme. See over here. So what I'll do, I'll just press Control and upper arrow. Now ATP is selected. So understand where we can see the surface, go to Actions, Surface and Show. So we can see that this is the surface of the ATP molecule which is getting interacted with this particular active site. So if you're not very clear with it, I can also change the color of the background. So you just go to Actions, Color, All Options, change it to background and white. So we can see that ATP is bound over here. Magnesium is very much nearby to it. We can select magnesium from here and change the color and show the surface as well. So, yeah. So now if we need to check the surface of the entire protein, which I showed in the presentation, the SARS-CoV-2 spike protein, I can show you the entire surface of the protein for this particular protein as well. So you just need to select all, go to Actions and show surface. Yeah. So this is the surface of this particular enzyme. So you can rotate it as well. You can do zoom in, zoom out from your mouse or touchpad. Now, if you want to see the active site where ATP was bound, what you can do, you can just clear selection, go for the residue that is ATP that has been selected and change the color. So we can see that this is the surface where ATP was bound, and this is the active site of the HPPK enzyme as such. So I'll just take the color as on. So this is the surface of the protein. Clear selection. Apart from this, we can actually remove all the surface and we can see what is the distance between two residues. So for that, it's important to remove the surface for our own understanding. And go to Tools. Now tools in this, we can actually make a movie also. We can do the structural analysis. We can structure comparison. We can check the sequence for the same. For the PDB has been uploaded. So we'll just check on the sequence. So this is the sequence and these particular residues are disordered. That's why they have not been included. But if I check and click on this, so this is the structure which this particular amino acid was made. So now if we check this one, so this is the one where our protein is starting from here. And we can see that these particular residues made a beta sheet over here. Whereas for loops, we can see this one as well. Or if you want to check the loop is making how many residues, you just select on here and press over here. So this selected it gets selected over here as well. So these are very basic things which you can actually play around, and unless and until you start using it, it will be a bit confusing for you. But once you get a hang of it, it will be very easy to play around with it.